Thank you all for joining. Let me. Yes, looks good.
Looks
Good,
great. Yes.
good. So I will try and give a bit of an introduction and discussion of the hologenome and holobiome concepts. And I'll first talk a bit about the history, and then we'll have some discussions and questions, and then give another short presentation about why it is controversial, and then we'll discuss again afterwards. So I thought a good starting point would just be to talk a little bit about the basics of natural selection. So we have variation in a trait that can be inherited. So this could be something as obvious as height in giraffes. We have competition for resources that leads to a difference in reproductive output. So here, if you have a longer neck, you are more able to get the leaves on the high plants. And therefore, we can see potentially an adaptive change over time in the populations. So here, there would be selection for long necks. We often see competition for a very particular resource, which is access to mating. And then we can have sexual selection results in these exaggerated traits that can be costly to carry, but increase the reproductive output. So the theory of evolution by natural selection is very much founded on competition and the struggle for existence. So that was one of the reasons why it was somewhat of a challenge to both Darwin and to others to explain how it is that we can sometimes see elaborate cooperation arise within a species and be maintained, such as the division of labor that we can have in social insects. So here it's a bivouac built by army ants, and all the workers are sterile and they're protecting their reproductive queen, who's hidden in the middle somewhere here. The problem with maintenance of cooperation was illustrated by this tragedy of the commons concept. So here we have some farmers who have a shared resource, which is land used for grazing of their animals. And as long as the farmers only put on the amount of animals that they've agreed on, the carrying capacity of the land, then all is good. But the problem is that the farmers, of course, can each gain an advantage by just putting out a couple of more animals. And then at some point, when everyone does that, or even if it's just one that does that, the common resource is, of course, ruined for everyone. We now know that the kin selection can maintain these altruistic or cooperative behaviors within the species. So here an individual gain inclusive fitness by helping its relative reproduce. So here we have a ground squirrel, which is warning its family group about an approaching predator, and as such helping its family members and relatives reproduce in the future. So it can, of course, be complicated enough to understand the selective pressures within an individual species, but it has long been recognized that a species don't exist in a vacuum. They also interact with other species very often, very closely. So already a few years after Darwin published his main work, then Debarry actually coined the term symbiosis as the living together of dissimilar organisms. Often we have a size difference, so we have a macroscopic host, so it could be an animal or a plant, and then we have some microbial symbiotic partners. And here's two of the systems that Debarry was working on was ectomycorrhiza of plants, so a fungus-plant interaction, where the plant produce sugars and the fungus produce contribute with phosphorus from the surrounding soil, and also the roots of here on the other side of legume plants that are colonized by rhizobia, bacteria that fix nitrogen for the plant. So symbiosis in itself is a term that has a lot of different interpretations, both at the time and now. Debarry used it to cover the whole continuum of interactions, from parasitism to commensalism to mutualism. Whereas now and then, a lot of people saw symbiosis only as mutualism, where there's a net benefit to both of the interacting partners. And this is to some degree just a question of which definition you prefer, but it also reflects some of the controversy in how these interspecies interactions has been considered historically and to some degree now. So the discovery of microorganisms was from the beginning very biased towards disease. So some scientists simply found it unbelievable that there could be any benefit to the host of harboring bacteria, for example. And then on the other hand, we had someone like the Russian Kropotkin, who was strongly biased towards this harmonic worldview, and that organisms within interactions within and between species should be dominated by mutual aid. And now, how most evolutionary biologists see mutualism is that it's a mutual exploitation to the net benefit of both. So more than a century of symbiosis research has both shown us a lot about the wonderful complexity we have of interactions and how fundamental they really are to all parts of life as we know it. A couple of factors have been found to contribute to where we find ourselves on this spectrum between parasitism and mutualism. So there are some things that can better align the interests of the host and the symbionts. So one of these factors is a mode of transmission, and we see that vertical transmission favors cooperation, as the symbiosis, or the symbionts, the microbial symbionts have an interest in the host's successful reproduction. But there are, of course, tons of exceptions also to this. Another factor is how dependent the partners are on each other, as you might, as a symbiont, be less inclined to harm your host if you're completely dependent on it to finish your life cycle. So the usefulness of viewing these interactions on this spectrum can also be reflected in how we can, for example, see how changes in the environment can shift an interaction along this spectrum. So we could imagine that if the environment, for example, means that there's more or less of a resource, then this changes the cost and the benefit of the different partners to contribute to their association. An example of how this framework can be useful to understand host microbe interactions, I would really warmly recommend to check out the work of Toby Keir's lab from Amsterdam. So they've actually looked at two of these symbioses that Debarry started his whole concept of symbiosis on. So the mycorrhiza and the rhizobia and legume interactions. And what they do really is consider the interaction as an economical marketplace, and where each partner has something to contribute, and the environment and the condition determines how much these different commodities are worth. And in their beautiful experimental setups, they can manipulate how much the different partners invest, and also how much their contributions to the interaction is worth. And then they can see what happens in the interactions when one partner doesn't deliver, or the price of a commodity, the value of a commodity changes in the environment. So we started out really with the focus on association between one host and a few or just one very specific symbionts. So this could be an interaction such as that between the squid and the luminescent Vibrio bacteria, where the bacteria are more or less grown in a monoculture and they produce light for the host. But now, of course, with the much more advanced sequencing, this has really revolutionized our understanding of how diverse microbiomes can be. And one obvious example is, of course, the gut of mammals. And here we can see there's a variation in the microbiomes between hosts and over time, and that this can be correlated with strong phenotypes that affect the host fitness. So what we can observe across a diversity of different systems is that the microbial symbionts, they add variation in function, and this may be heritable, and the host and the symbionts are more than the sum of the parts. And thinking back to the giraffes and these different elements, you can see that this is pretty much what's required for natural selection. So the prediction is that natural selection can act on the level of this association between the host and the microbes. And this is the foundation of the Holobiont and Hologenome concepts. And part of the beauty of this is, I think at least, is that multiple scientists have arrived independently at this idea based on what they've observed in their different experimental systems. So I've attempted to make a bit of a timeline with some of the key players and don't put too much attention into the years and the names specifically. But already in the 40s, the holobiont term was coined by a German scientist, and he really used it as to describe the different partners in a holobiont where the in a holobiome where the holobionts were completely dependent on each other. More well known is Lynn Margulis, and she introduced the term independently in the 90s, really describing how the holobiont is a so the symbiosis is a source of evolutionary innovation. And shortly after, and independently, I think, Richard Jefferson introduced the holobiont and the hologenome, and his focus was really on that it doesn't make sense to talk about macroorganisms as independent of the microbes. So his work or his thinking was really based on how microbes are essential to the hormones that hosts produce, and hormones are essential for reproduction and mate choice. And in that sense, microbes is really what's driving reproduction in hosts. So it doesn't really make sense to even talk about hosts, because they wouldn't be what they are without the microbes. The Rosenbergs a bit later also started talking a lot about holobionts and hologenomes. So they were working with corals, and the core of the coral symbiosis is, of course, the zooxanthellae and the animal host, but there's also lots of other microbes living in the mucus on the coral or in the coral. And what they experienced was that the microbiome of the coral could change over a relatively short time scale, and that could contribute to disease defense. So this was a way to have adaptation within the lifetime of the host. And they also really see this as an organism, the holobiont. A couple of years later, also in collaboration with the Rosenbergs, we have Bordenstein coming in and writing a couple of seminal reviewed papers and really introducing the holobiont and hologenome as a biological unit, and there's potential for selection at all the different levels, both the microbes, the host, and then the association as itself. And then, of course, here at the foundation of the center, with a slightly somewhat different take on what a hologenome and holobiont is related to the Bordenstein definition, I would say, where we're just interested in what the macro and microorganisms are present, and we don't necessarily imply that there should be selection at the different levels. And what I see is particularly important is that we're bringing the host, the variation, the possibility, and the importance of variation in the host genome back. So just to wrap up, and we'll get back to that. The earlier definitions and use of the hologenome really stress that what's most important is the selection is working at the holobiont, the hologenome level. So when we look at it all together, that means at the host level, we have Mendelian inheritance, we can have recombination and mutation across generation. But then when we add the microbiome, a lot more can happen. So we can have a horizontal gene transfer between microbes, and of course have mutation. We can also have loss and gain of specific species of microbes, and we can of course have change in frequency that can all matter. And this combination of the host and the microbiome can affect behavior, sociality, nutrition, and defense. And in that way, it can contribute to acclimatization, for example, in terms of when the environment is changing, such as climate change. And it can also lead to adaptation and speciation, even if, for example, the microbiome contributes to mate choice, which has been seen in some species. So that means that we have an interaction between the host genome, between the genomes of the microbiome, and then the environment. Going back a bit to Bordenstein's and associates definitions, so they're really stressing that complex multicellular eukaryotes are not autonomous organisms, and that the hologenome consists of both the nucleic genomes, the organelles, and then the microbiome. And that we can equate the microbes as genes in the hologenome. The microbes can be compared to genes in the host. The term holobiont has been used a lot recently, so it's gained in popularity. You can see in terms of how often it's used in publications, but that doesn't mean that it's without controversy, and that's part of what we'll get to next. I have some suggestions for discussion points, but I'm thinking we can maybe first see if there's any questions or comments to this first bit before we go out in breakout rooms.
I kind of think there is a major problem we faced, which is there are people that expect there to be a very clear, precise definition of what is a holobiont and what is a hologenome. And to me, it is all context dependent. It all depends on what the question is you're working on. So I like to view it as this kind of fuzzy idea. We know approximately what a holobiont is. It's a union of microbes and a host, but exactly which microbes are in union with the host and which of those microbes are important very much depends on the question of interest. So for example, to give the extreme example, and I may have this wrong, and Sandra should be able to correct me if I do, I'm pretty sure that, in fact, I'll use an example I do know. So it's quite well known that some of the worms that infect humans are detrimental in general to humans, except it also turns out they keep your immune system not quite as active and don't give you allergies, right? So whether the pigworms or whatever it is, the whipworms that humans get, should be viewed as a holobiont kind of depends on what is the question. Are we looking at what helps protect us from allergies, or are we looking at what are the things like reducing human health in Guinea-Bissau or whatever, right? So one can think about physical and temporal boundaries, but I think, ultimately there are many microbes on us. Sometimes we have microbes that are irrelevant for us, but they're important for other people. Other times you have microbes that vanish and never go. And that's kind of why it's hard for me to imagine there being a concrete definition, right? which is why it's hard to imagine the concept being under selection. If you can't even define the concept, how can it be under natural selection? But again, if you come back to this thing I talked about before about if it is just the phenotype, then of course one looks at the relevant parts of the phenotype, depending on the question, and then it kind of makes the problems go away. But I know a lot of people, especially the people who always criticize whatever I say, hate this idea, but they're the same as the people I fought with for years in the bird community who bird systematists love to have a bifurcating family tree of birds. And as we sequence genomes, we see that lots of birds are hybrids. And they're like, yes, but then what does that mean about the relationship? And the answer is, well, it's not a bifurcating relationship. And they're like, yes, but how do you get a bifurcating relationship? And we're like, well, you can't. And you kind of go around in this fight because there is an unwillingness to accept an alternate model. So anyway, but again, the reason I brought this up is with your second point, an opportunistic pathogen may or may not be, depending on if it's being detrimental or good. That was, yeah. Anyway.
Yeah, no, but I also think that it doesn't have to be good to be part of the holobiont. It's just it has to be there and be relevant to what you are.
Yes, relevant is the key word.
So it could be like this equivalent to a neutral genetic but variation, so that it defines your adaptive potential when the surroundings change.
Well, yeah, yeah, exactly. It could potentially be relevant, right? And then the question I think that we're mostly interested in is when are certain parts of it relevant and what is driving that need, right, for it to be relevant. But, you know, so, and I want to mention another thing I told the people in my group. One time I got, in fact, 3 times now, I got the same question phrased in different ways. And the person said to me, okay, you are saying that whatever the microbe is in a cow that digests grass is critical to allow it to eat and get nutrition, right? And I'm saying yes. And then he goes, well, what about the eucalyptus that a koala eats? The koala cannot exist without the eucalyptus. Is the eucalyptus part of the holobiont of the koala? And I've never really had time to think about this or give a good answer on the spot, but I get their point, right? How does the microbe that allows the koala to eat the eucalyptus differ from the eucalyptus? Because in both situations, it's screwed without it. It has no other option of what to eat. And
yeah, but I think at least also if you go to the Bodenstein paper, then the eucalyptus tree is one holobiont interacting with another, which is the koala.
But then why is the microbe in the gut of the koala not a holobiont interacting with other microbes in the gut of the koala?
What?
Yeah, but then how is that different to the microbes in the gut of the koala that need the other microbes in the gut of the koala? They're different holobionts.
That's a confusing question.
No, but that's the point, right? I mean, it's like these they're kind of fractals almost,
I think,
right?
isn't it at this point a matter of how we define, and maybe not defining it, like Sandra said, defining based on the question and the relevance to the question that you're asking is a better way to go.
I think it's the only way to go.
Yeah, yeah.
It is the only way to go, but you can see why people get upset with the question, is the holobiont under natural selection? If you can't define the holobiont, then of course it makes it very hard to answer that question, right? Because
yeah, but I think it's also like there's lots of traits that characterize me, but not all of them are going to be under selection for. I mean, there's lots of things that make me, but they don't necessarily contribute to my reproductive fitness.
Yeah, it just depends on the context, right?
But I guess also depends on the first question that we have the physical boundaries. So if you take koala and isolate him, his own microbiome would be with him. But yeah, but not the eucalyptus. It's like also the physical boundaries where you set. So I mean, I guess if we answer the first question, we kind of answered that one as well, maybe.
But the question is that a useful definition? Because basically, if you put the koala and its microbes together, what does it mean? I mean, it's not going to survive without the eucalyptus. So it's
yeah, it becomes like the whole ecological thing as the whole meta. Yeah. But I guess it's just the physical size and it has to be a microorganism or something, and that if you take the organisms
right, I don't
away.
know. Look at, is it lichens? Is it which is the one that's the famous plant-like thing that's like a hybrid of two things? Lichens?
Lichen.
They're not microbes, are they? Aren't they like a...
It's,
Algae and fungi.
Algae, yeah.
Well, it's cyanobacteria, right? So it's
I don't know.
Yes.
almost microbes, and then a fungus.
As you can see, I've got a very, very grip of biology, which is ironic, really. So Sandra, your question about do you see metagenome and hologenome, I have an opinion on that, but I'm quite curious to hear other people's opinion on that. If somebody would like to share an opinion.
As you can imagine, we didn't reach that question.
So I have a, can I go back to the first question for 1 second?
Only if you answer the third question first.
Yeah, I think that's related. I like the idea that Morton brought up of discussing what a meta-organism is. And I think I, for some reason, can better relate to a meta-organism being a separable individual, like a koala is a meta-organism. I can live with that. And I think then a meta-organism has a metagenome, a holobiont has a hologenome. That's easy.
Yeah, but if you take a gut sample from a human and shotgun sequence it, is the metagenome just the microbial fraction or the microbial and the human fraction?
As a matter of convenience, the microbial fraction, but as a matter of utility, the entire thing.
And so what is the hologenome then?
And I don't want to define a holobiont, so I'm not going to tell you what a hologenome is.
I mean, I would argue the hologenome is just the fraction of the metagenome that is relevant for your question.
Yeah.
But I don't know if that's right. That's an easy cop-out.
But I think that's a difficult definition, right? Because that is an a posteriori definition. You can only define it after you've answered your question.
That's not necessarily a problem. It's, if you're saying we're going to go out to identify the hologenome that allows the vampire bat to drink blood, I mean, that's kind of like saying we're going to go to a forest and find what keystone species are in it. We've said the word keystone species without knowing. The study is revealing what the keystone species are.
No, I agree, but it's not useful as a definition, right? Because what does it tell us?
Well, it is, because if I then want to say, I now can tell you what the hologenome of a vampire bat is, I can list the key traits.
But then it depends on what you're defining as the key trait of the vampire bat, the ability to eat blood. So it comes with some phenotype that is, okay.
But that's the same as the holobiont then. The holobiont comes with a...
Yeah.
I mean, so just to go back to terms, I'm sorry, Sandra, if you have to move on, let me know. And if anyone wants to interrupt, just shout. So Richard Jefferson, who you mentioned, who some of us had the pleasure of meeting, he has these three terms. He has this term holobiont, merabiont, and apobiont, which are quite useful for thinking about. So let's forget about the holobiont for a minute. The apobiont he defines as the scaffold. Basically, you take all the microbes off the bigger thing. That is your apobiont. It's just the microbe-free thing that can't exist. The merabiont is actually a kind of minimized version of the microbes on that scaffold that allow it to exist. Maybe not very well, but it kind of needs this reduced diversity. I mean, it can survive with the reduced diversity. And the holobiont is the more natural diversity, right? Now, actually, holobiont is difficult to define, but apobiont can be defined as the thing would be dead without them. And then, of course, ultimately, you could like make a list of the minimal required microbes to exist, maybe a very crappy existence, right? But I guess the point is, the more, everything on top of the apobiont is like extra benefits you could have that could make your life better, and maybe a load of extra random stuff, right? But the apobiont is kind of, you cannot exist without that apobiont, right? You wouldn't be alive, right? And then, so it's like the minimal basic thing, like when you buy your computer and you have the minimal basic thing before you put on all the extra fancy bits of the Danish keyboard and stuff. Anyway, I think they are useful terms once I actually understood what they meant. So,
so in that case, the eucalyptus will be part of the holobiont as well.
Well, that goes back to that complex question again. I mean, but then again, what about oxygen? Is oxygen part of the holobiont for us humans? It's not life, so I guess.
If it don't live, it don't count.
Yeah. Are plants part of the holobiont for humans because they make oxygen and we die without it? I mean, it does get very complex at that level. Anton, sorry.
No, just one comment about the metagenome and hologenome definitions. I think that's how we use them, and I think it's good to for the sake of organizing stuff, as we are usually dealing with one host organism, a community of microorganisms. We usually define the host genome and then the microbial metagenome. So I think we can define as we want metagenome, but for the sake of splitting and defining two elements that we know have very different biological properties. I think it's a nice distinction of having the host genome, microbial metagenome, and all together we define the hologenome. That's how we usually use it. And I think it's good enough. And we don't need to, to discuss much more about that. Just after saying that this is completely arbitrary. So we define it, but we could use other terms.
I agree, but at least having the discussion prepares you for what other people will bring up, right?
Yeah, of course.
What's important is to be able to argue why it's not really a problem or why it can be flexible. So, and I would
for me here is the individual and the community. These are the two key things for defining one as a genome and the other one as a metagenome.
So in your case, so when you say the individual and the community may need not necessarily all be good things.
Of course, yeah, but the same as your genomic traits, not all are good for you.
Someone come up with some of their favorite examples of a holobiont or hologenome.
Yeah, the vulture, that's my baby.
Someone else than Tom.
Maybe Andersen can explain it better than I, but apparently there are these newts that have these toxin-producing bacteria, and the way they can tolerate them is that they have some mutations, variants in the newt's own, the host own genome, that makes their own natrium channels resistant, so that they're like, what do you call it, resilient to these toxins. So it's like a clear adaptation in the host genome for being able to host these toxin-producing microbes that are then keeping all the potential predators at bay. And I thought that was a pretty neat model. Was that correctly interpreted, Andersen?
Yep. It's a new paper in eLife Money. It's from April this year. I can copy paste it in the chat. It's a really cool paper.
Yeah.
So can I, however, ask Anton to say the example he told me by email the other day that he's put into the paper about the immune system, and I'll say why afterwards. You know, Anton, you wrote me an email the other day.
The immunoglobulin one?
Yeah.
Yeah, so I think, well, I don't know if it was this one, but the fact that there are some bacteria and only some bacteria that use immunoglobulin A, that they bind to immunoglobulin A so that they establish in the mucosal barrier of the intestine. And it seems that these same bacteria are the ones that exhibit the largest heritability. Because depending on how these immunoglobulins differ, depending on what's the genetic structure of the host, then some or other bacteria can be attached to those to the mucosal barrier, and then the rest of bacteria are kind of the secondarily or indirectly affected. So first, these bacteria that bind to the immunoglobulins are affected by the host, and then depending on how these bacteria are filtered, then this has a downstream effect on how the rest of the community develops in the intestine. In a simpler way.
So the reason I wanted to highlight that is, again, it kind of underscores the different viewpoints of hologenomics. So the vampire or the vulture, all these things, what we're seeing is basically an explanation of how the microbes and the host work together to fix a question. But what Anton's talking about is how actually host genetic variation has a very consequential effect on the microbes, which in turn has a consequential effect on what's going on, right? And the reason I bring this up again, although it should be clear by now, is because one of them is to some degree stamp collecting. It's just, here's another example, whereas the other one is a fundamental mechanism, right, showing that how host genetic variation can shape things. And I think this is kind of important, because when people ask me, like, what are you really looking at? Are you guys just cataloging yet another example of what's going on? My answer is no. We're more interested in the underlying, how is the host variation shaping it in the first place, right, and driving the system. So it's the kind of two extremes of this, going from just yet another example. And the reason I say this is there are lots and lots of papers coming out about holobionts, which literally they will pick a new system like the newt thing. And so here's another example of how life wouldn't work without both. But there are very few papers looking at, okay, but what happens when you vary the genetics in the host to the actual system and so on. And so to me, that's where the real evolutionary hologenomics comes into.
Okay, so as you probably gathered by now, it is, even though we can see why it all makes sense to talk about hologenomes, it is controversial. So what's all the fuss about? Well, I'll start with what the fuss is not about. It's not that people don't think that microbes are important for the host. So some of the biggest opponents of the hologenome concept are very well renowned scientists who spent their whole career working on specific symbionts of insects, for example. So it's not that they don't believe that the microbes are important. It's more whether selection actually acts at the hologenome level. So does it, and is the hologenome ever the primary unit of selection? Or is there too much conflict within the microbial community and with the host and the microbe for the hologenome to be a unit of selection? Part of this, of what would determine whether selection can act at the hologenome, holobiont level, is whether the microbiome is heritable and whether it can impact reproductive success. So we have lots of good examples from, for example, microbes associated with insects, where we can really trace how they're transmitted through generations and where they're found at all times. But then, of course, we have much more messy situations, such as the gut microbiome from humans, where there definitely is some heritability, but how strong is it? How much of the microbiome is actually inherited, and how much is later added from the environment? And one of the concepts that has been introduced in favor of the holobiont is phylosymbiosis, to argue that complex microbiomes can be heritable. And so this is also from Seth Bortenstein's group, where they show that microbiome community composition matches the host phylogeny, and they've done this for lots of different host microbiome combinations. Some of the opponents argue that this is studied at a too coarse a scale, so that they look at 16S to compare which microbes are found in which hosts. And you can't really say anything about whether microbes are actually closely related from just 16S data. Then there's also a question of whether host and microbes are actually co-evolved or just co-speciated. So whether there are adaptations in the genomes of both based on the relation, or have they just kind of followed each other. And then there's also especially the question of microbes that are not only host associated, but also found in the environment. How important is fitness in either the host or the free living environment? Where do they gain most of their fitness? Because this would severely limit the potential for evolution, co-evolution with the host, if the microbes spend a significant part of their life in the environment, such as, for example, the case of Vibrio bacteria that are associated with the squids. I really like this this review opinion paper on the microbiome on a leash, where we can kind of think of different levels of potential for host control. So when we see microbial traits that are beneficial to host, we tend to assume that this is kind of the result of co-evolution. But what is actually the potential for co-evolution, and how much can the host control its microbiome? And so they put up these different types of association, and the squid Vibrio association is kind of how we like to think of a situation where the host really is in control of the microbes. So the microbes have to express specific traits to be kept by the host, and the host can expel microbes as well. And then we have my beautiful zombie fungus here that takes over the ant host. And here's obviously that the symbiont or the parasite controls the host. And then we have something like the gut system, which is something that a system that a lot of us are interested in, where we have a complex microbiome. And here it's of course what the microbes are experiencing is interactions with a lot of other microbes. And because the microbes have a short generation time, what is important here is how they are doing in competition with the other microbes in the gut. So how can the host influence and control the composition of the microbiome? And some of the mechanism that has been proposed is that the host can control the resource availability. So this is, for example, through what the diet is, but also what's secreted that either benefit or moves away microbes from the system. And we can have a compartmentalization that separates types of bacteria from each other, or that so that of course only specific tissues are available for the microbes. So one of the big points from the critics is also that host dependence on microbes for their development does not require co-evolution between the microbes and their host. So in this sense, you could say, for example, that the development of the mammalian gut is dependent on cues from bacteria, and we don't have normal development if these microbial cues are not available. But that does not mean that the microbes have evolved to be sending these signals to the host, but the host is rather using these microbial cues because it's a reliable source of environmental variation. So this whole controversy of using the hologenome is really about the interpretation of whether the hologenome, the association, is what is the primary unit of selection. And if we don't assume that, which is, for example, in most of the Bordenstein papers, it's that we have the potential of evolution to act at the hologenome level. It doesn't have to be the most important in all situations. So the controversy is really, to some degree, semantics. What you whether you interpret the hologenome as meaning that that it has to be the most important unit of selection, which is not what most of the people actually use the term, how they use it. So I think what has happened is that we've gone from a very host-centric world where we don't even really consider the microbes to a very microbiome-centric world where we have had some sort of expectation that everything can be explained by microbes. And we expect to find that any sorts of disease or healthy state can be attributed to microbes. But we're at a stage where we still don't really know what is a healthy microbiome when it comes to humans. There's so much variation, and what's health in one healthy person is not necessarily what will make another. We also cannot answer a question like, does probiotics work? Why does it apparently sometimes? Why doesn't it in other situations? And can we actually manipulate microbiome composition in a directional and a meaningful way where we can predict how it's going to change? So what I see as one of the beauties of the center is that in the way that we're using the hologenome concept and holobiome concept is that we're bringing back together the importance of considering host variation and microbiome variation, not necessarily saying that that they're both always important, but that it should at least be considered, and that variation in either can be beneficial, neutral, or harmful, and especially that the effect of variation in either may depend on the environment. So that, for example, if you have a very controlled environment, host variation may be important, whereas in a situation where we have a lot of variation from other factors, it may matter less. And then there's a couple of more discussion points.
We actually would like you to explain the second question. That was our conclusion.
No, the third one. The third one.
There's
Oh,
only two questions.
Yeah, the second one. Sorry.
Yeah.
Yeah. So what I was thinking was, if you, so for example, if you have a competition within between microbes within the gut, for example, so that this results in a less than ideal production of some nutrient for the host. So there is some conflict within the holobiont.
But isn't that when you have conflict between paternal and maternal alleles in a host? I mean,
Exactly. Yeah. No. So I would say that no, you can have conflict within the holobiont and still have selection at the holobiont level, because even if we don't even considering the microbe, but just considering our own genome, then there's still conflict, and we still. Does that make sense?
Yeah, I guess for me the key words in both these points are the word potential for and possibility for, because I think this is a part of the problem. There is, as long as you accept that we're looking at potential as opposed to actually happening, then it's very easy to discuss. The problem is when people are out there saying it's impossible, no, you can't have it, or it doesn't happen always, therefore it doesn't happen, right? So I guess, yeah, it's, and your first one about times, which we were discussing, it's like, I would imagine a microbe in association with a host for its whole life has got a greater chance of being under selection than one that's got a very, very short association, right? Because that's just probability. Having said that, short association at different times of life can have massively different potentials, right? If its association is for doing whatever Richard Jefferson says about digesting down sex hormones and whatever, right? Massive chance, even if it's only there for a week.
But then there's a potential for a very strong host effect. But if the microbe has a very short association, then what it's doing has likely nothing. It's just a part of its microbial metabolism. It potentially has nothing to do with what it does to the host.
Yeah, so you're saying it's not the strategy of the microbe's life to be there. That's correct.
Yeah, which I think is also really the whole gist of the microbiome on a leash paper, is that we see that the host has co-opted all these things that microbes are anyways doing when they're interacting with each other and when they're metabolizing compounds.
But so obligate gut microbes, right? I mean, I think there are some, or in some systems, there are some microbes that can only exist in the gut of the animal, right? Is it possible to say that is not selection on the holobiont? Is there a way to argue that that is not actually under selection? Because to say,
but I think you can still have interactions within the microbes that are, for the microbes, is only about whether they can compete, how they're interacting with other microbes, and that can still, so they are producing compounds based on their interaction with other microbes.
But if
that
they
has
can't,
an effect on the host, and that the host
if they can't exist without the host gut structure and the conditions, they're not independent of the host, right? The question is whether they shape the host in that way or what? Is that what it comes down to?
Yeah, but for them, the host might as well have been a lake, or
I guess it's just static, right? The host is not, they're not affecting how the host is transmitting its
But
genome.
the host is actively fighting out other microbes that will perturb the system. The host is producing nutrients. The question is, did the microbe shape the host to fight out the competition in the first place, right? So it's that, is the host driving it at all? I mean,
and there I just think that because of the generation time of microbes, the interactions with other microbes is going to be so much more important for the majority of things, but not all.
But just because it's more important doesn't mean it's not important. So
no.
But the critics are saying it is not under natural selection. They're not saying the effect of natural selection is much less, right? It's very much, it never happens. And I guess the question is it an impossibility? Can it never happen? Or is it just not very common? And that's the same as genome versus environment shaping the microbiome. The reason people discount genome is because they say the environment's so important, the genome is irrelevant, which I would strongly argue is wrong, right? But I can see their point. But there's a difference between a small effect versus no effect. And I guess when I read like, what's her name, Nancy Moran's kind of replies, they're like, no, it's an impossibility. There is never natural selection at that level. And I guess that's what my query is. Is that right? Or is it?
I disagree with that.
Yeah. So that's similarly why I disagree with the fact that host genome is irrelevant when, you know, it's a wording thing, right? And it would maybe be useful to find some examples of where absolutely it's very hard to explain anything without there being natural selection because the things are so tightly intertwined and they have to have that certain thing, but that may be impossible. Yeah.
Yes, what we thought was intriguing about this first question was that it was pretty easy to start talking and come up with cool examples where clearly the host is selecting on the microbe community, like the newt example, or vice versa. But then if you look at the last 5-6 words of the questions, is selection needs to act at the hologenome level, then it just illustrates how more complex the whole thing becomes.
But a lichen is surely under natural selection, right? Or is a lichen not under natural selection? I mean, can the two parts exist without each other, and are they not shaping each other?
I mean, there's papers on lichen selection, so I'm pretty sure it can.
But if a lichen is a holobiont, which is the host and which is the microbiome?
But does that matter?
Yeah, that doesn't matter.
But what I think someone like Nancy Moran is arguing that even when we have, even if you have these extremely obligate association between an aphid and its bacterial symbionts, that there's still what makes you ask the interesting questions is the conflict between the host. Where are the conflicts and where can we see that expressed? And what are the adaptations from the microbe side and the host side, and so it's, yeah, no, I don't know what I'm saying, but it's just that you look at the individual parts and you don't get anything extra by considering the two together.
Now that's a very important statement. You don't get anything extra by considering together.
Yeah.
But isn't that the underlying concept of the center, that looking at both of them?
Oh, you muted yourself.
I'm very good at this, apparently. I've done this a couple of times. So I was just saying, isn't that the underlying concept of the center, that looking at both of them gives us more than looking at each one of them? And also adding to what Martin was saying before, it actually was quite easy to think of how the host, much of the discussion we focused on how the host might affect or select on the microbiome, but it's also important to think of the other direction, right? About how the microbiome might be selecting on host, and that's how you're transmitting any of these host genetic changes or variation that's adapted to the microbiome or beneficial to the microbiome forward. Otherwise, like Sandra said, it could easily just be a lake bed.
To me, it's just easy to see how selection acts on a phenotype that is the sum so of the two. So it's a holobiont.
Yeah.
External that is affected by selection, but the actual heritable part that's selected for is either in the host genome or in the obligate microbiome or bacterial genomes, but it's not the both. So I just have a hard time to envision how you have selection directly affecting both parts. Or if it's fine that if it's only selection on the host genome's ability to tolerate or culture a beneficial microbe, then that's also part of the hologenome, even though it's only the host genome variant that is selected for.
But I guess if we look like evolutionary wise, right? For example, let's say we have the koala again, example, then you get this digestive amoeba that makes eucalyptus. Then on the longer run, this affects the koala to become a koala, which is an animal that climbs the the tree and becomes more adaptive to that eucalyptus environment specific. I mean, basically, if the initial environment forced the gut to change or to favor those specific microbes to evolve, then the microbes force later on the koala to, to get that niche and become adapted to that specific. And that basically what ultimately makes koala, right? I mean, just the feeding on, I mean, may take longer, but it may be eventually affecting. I don't know.
Just one question to this: do the selection, when it's is it imagined that the selection has to be ongoing both at the genome and the microbes at the same time? Of course, over whatever generation times we're talking about, or as you with like describing there, it's like first we have some selection going on maybe the microbiome treating, and then it's changed that so it's heavy selection on the host. And but how is it in this perspective? Would that be counted as two separate selection events and has nothing to do with a hologenomic selection? Or
I think part of so that the Bodenstein argument, at least, is that because you have the genome where that can change on a whole different time scale, this opens up a whole new range of possibilities for the host or for the holobiont, so that we have adaptation at different time scales. So what has often mentioned in these reviews is that you can almost say that it's sort of a reintroduction of Lamarckism that you can change in your lifetime. So that Lamarck thought that the giraffes would be stretching for the leaves, and then they would grow their necks, and then so this trait could then be passed on. And this is kind of what the hologenome does, because you can have the variation, or as I think you nicely put it, Anton, in one of your reviews, that you can call maybe this acclimatization on a shorter time scale that can then be passed on and lead to adaptation in the longer run.
But that's basically the same as epigenetics, right? Mm. Which is also Lamarckian to some degree, I guess. Yeah. But I can see why people don't like that.
But then that would have to lead to adaptation, real adaptation in the genome later, right? In the host genome later.
Potentially.
Not necessarily.
All right. But wouldn't that be a requisite? Because it has to be selection on the genome, right? For if it has to look at the hologenome.
Well, isn't the whole point that it's not selection on one or the other, it's selection on both at once. Yes. Without both of them, you can't have the thing in the first place. So that's the kind of implicit assumption that the selection is happening on both parts altogether.
But then on the microbes, is it, because some of the thoughts or some of the things you said, Sandra, as well, is that the microbes can live a lot of other places, but still, that I'm just thinking, even though there's selection in the gut or the different places in the hologenome on the microbes, and they evolve, but they can still live other places even though they are under selection while they have this.
Not always.
Okay.
I think that's one of the arguments why you wouldn't have the hologenome as a unit of selection, because it's such a small part of the lifestyle. But there are obviously microbes that are only found associated with the hologenome.
But is it a prerequisite, you could say, that the microbes end up not being able to live anywhere else than together with the host?
I don't think it's a prerequisite. I think
it makes it more likely.
Yeah. But a requisite of it being under selection is there has to be at least some obligate relation. Not obligate, but I guess. Yeah,
I think maybe something like the Vibrio bacteria with the squid that have again, it's very difficult to quantify how much of the life is actually spent outside of a host, and how that how important that part is for reproduction. But there you still have very specific adaptations, right? Even though they're not strictly host associated.
A couple of comments. I need to leave in 2 minutes, and I might have missed something because I was preparing stuff for the kid. But two things. One is related. I don't know if you have also already discussed about the functional redundancy of the microbiome regarding to this holobiont level selection, because there might be different microbes that are providing the same service to the host, and that happens quite often. In the newt example, for instance, that Morten mentioned, there's not just one bacteria that is producing this toxin. There are multiple. So that is one thing that to consider. And the other one is that the microbiome is a community. And sometimes we refer to the microbiome as if it were an organism. And if the evolution of the microbes is occurring also at the organism level, at the individual level, then sometimes I think that there are some mix-ups about how to treat evolution when we are comparing one organism against a community. And that's the thing, that there is a very clear mix-up sometimes with phylosymbiosis, for instance. What does phylosymbiosis mean? So we are comparing a community with a phylogeny of an organism. So we are comparing two different things. And I think that very often there are some mix-ups about.
Absolutely.
Yeah. And now I need to leave, so I'm sorry. I need to pick up the kids now.
Adios.
Bye.
So I would like to highlight, I mean, what is very clear from this is it's very complex, but I would like to highlight that I specifically, when I wrote the center, sidestepped the whole issue because I knew it was this complex. And I do find it very interesting, but I also think it's largely irrelevant for everyday what we're doing, right? One doesn't need to invoke natural selection to explain if a certain genetic variant allows a certain fish to have certain microbes to be invasively selective, right? And that's why it's so successful. And that's why the center is called the Center for Evolutionary Hologenomics and not the Center for is the holobiont under evolution. And I would like to make that, but this is a problem because this is what the reviewers or the critics or the people listening mix up, right? So it is very important those listening today realize that there is this very philosophically challenging question of what is the holobiont and is the holobiont under natural selection. That is not what evolutionary hologenomics is. Evolutionary hologenomics is how is a host and microbe allowing evolution to happen, but not the mechanism or the exact term for it. And I think it's important to have that argument if somebody comes up and jumps on you and says it's stupid because Nancy Moran said the holobiont is not under natural selection. Might be true, completely irrelevant to what most of us are doing. Not to say that people can't start looking into this philosophical question should they want to think very hard about it.